Hello, this is Barry Morn from Trinity College Dublin, welcoming you to this bite-sized bio web seminar. Today's presentation is entitled Time, the Forgotten Parameter in Flow Cytometry, and is being presented by Dr. Alfonso Blanco. Alfonso received his Ph.D. in biology from Universidad de Vigo. He is now the Scientific Director of the Conway Core Facilities and Director of the Flow Cytometry Core Technologies in University College Dublin. He holds many additional positions, including co-director and founder of the Irish Cytometry Society, and is a councillor of the International Society for Advancement of Cytometry. As always, we will have a question and answer session after the presentation, so please type any questions that you may have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Alfonso at the end. So now, over to you, Alfonso, for the presentation.
First of all, thank you very much for the nice introduction, and thank you to the organizers for this opportunity. So, good morning, good afternoon, or good evening, depending on where you are following this webinar. As Barry has mentioned, I'm the Scientific Director of the Core Facilities, and mainly working for flow cytometry. I'm also, and I have the honor of being selected as one of the ISAC scholars, and I'm also the head of Excite in Europe. So the idea of this webinar is basically to emphasize the importance of one of the parameters that we have in flow, and it's underestimated if it's not unused, and it's basically the time. So the idea for this seminar is to give you some examples about how to use it, especially in our favor, but also I will try to explain how to monitorize by flow cytometry some of the physiological parameters, some of the physiological changes that are happening in the cells practically in real time. As you can see, mostly of these applications I will show will be physiological changes in mammalian cells. The only reason is because they will be the ones that we have widely used. But obviously there are many other applications that can be done in platelets, bacteria, or even in nanoparticles. So this webinar will be a little bit of a taste of what can be done and trying to open your imagination to what you can do. As we usually say, your imagination is your limitation. There are many parameters in flow cytometry that we can measure, like intrinsic characteristics like size, shape, the granularity, or if you're working with plants, you have pigments like chlorophylls or carotenoids. But also we have to consider the autofluorescence of your cells, which can be modified by the treatment that you're giving them. There are other extrinsic characteristics like the DNA or apoptosis, necrosis, internal, external receptors, physiological parameters, etc., that we can measure using different dyes, and this is where takes place the selection of those dyes. There are other things that we can analyze as well with flow, which are cell or particle concentration. And finally, we have this other one that, as I said, is one of the unused parameters in flow, which is time. So when and why we have to use time? For example, we should use time, and we are using time to check the state of the instrument during our quality control. It's extremely important to maintain the instruments in good conditions, and for that we look at laser alignment, we look at photomultipliers that are sensitive, or we can look at the dirtiness or the cleanliness of the instrument. I'll show you just a couple of these examples. So you have on one side one group of beads that has been analyzed, and the other one are the same beads analyzed after 2 minutes of cleaning. As you can see, it's a big difference, just for 2 minutes of liquid. This is something that we will do, but we don't consider the time here. We just run the samples and that's it. What we will need to do afterwards is trying to monitorize the state of the instrument, and that's why we use the Legendre curve, monitorizing if there are any changes in the laser, in the photomultiplier, the sensitivity, etc., etc. So again, here the time plays an important role. But when you're running the actual sample to something that we want to check, as you can see here, we have scatter versus time or fluorescence versus time. So at the beginning of this sample, you can see that it's quite strong here, the number of events that we have. But suddenly, as soon as the time flies, you can see that these lines disappear, became a lower number of events display. And the only reason for that is because the cells started to settle down after a while, so we are starting to drop down the number of events per second. In this other example, where we are showing again scatter versus fluorescence, when we display the time, you can see that the analysis is not as continuous as we could think at the beginning. There are some gaps, like in this case or this case, and you can see how the data has been modified and the coefficient of the variation can change on the time. And the reason here is because we have a clog on the instrument. We have some debris that are affecting our and finally we have this other one. You can see again the number of events is constant up to one particular point. Now the number of events increases, and again they increase. Again, on the fluorescence we can see the same thing. And this is again another question of time. In this case, basically the user, what he was doing was a bit of rush and was just increasing the flow rate. By increasing the flow rate, you have more variation into your data. So let's keep moving on. Let's see what we can do. Mostly of flow cytometers that we are using at the moment are pressurized systems. So that means that when you put the sample is pressurized, takes a while to get the right pressure and move them inside the flow cell and get the analysis done. For example, here we have CSNT beads that we have run, so you can clearly see there are 3 peaks. But also you can see some other peaks down here or down there. We don't care. We get the files and that's it. But when we look at the time, you can see this peak with high number of events. And this is because the sample, when you get the sample pressurized, hasn't been stable. It is stable after a few seconds, where you can see more or less the same number of events. So what happens if we just gate on that particular moment where the sample is not fully stable? Look at the profile. Okay, what if we just gate on the other side? Look at the profile. Can you see an improvement? I'm sure you can. So it's quite important to look at the time and just do the analysis when the sample is stable. Okay, sample should be stable here. Let's keep running my sample. Oh, it doesn't look as nice as before. What's going on here? Let's zoom out a little bit on the time and you can see a big deep gap. And this is because the user hasn't realized that after a while run out of the sample and what you have is air bubbles. As you can see here, when we gate on the right time, this sample population is exactly what we wanted, and after a while the sample is clearly not what you want. If you don't look at this stuff, obviously your data will not be as you expect. So use the time for gating out. Now this is a video, I will copy this from a friend of mine. He is Ree Gardner. He loves this video for explaining how to use time delay for sorting, or how to explain time delay for sorting. Basically this group in Denmark has built up a sorter for chewing gums. Peter, he doesn't like his chewing gums with one particular color. He buys these chewing gums in a pack where you have a mix of white and blue. So when he goes to the cinema, he doesn't like to get the blues on the mouth. So he decided to make a machine to separate them physically. So just to show you with the Lego pieces, he has the chewing gums, put them inside the machine, and start to collect the white ones on one side and the blue ones on the other side. It's so clear. Obviously, this is not what we are doing, but this is a representation of what we are doing when we are sorting. So time is extremely important parameter when you are doing cell sorter, not just when you are doing analysis. So it's extremely important to perform everything considering time. As I said, I will show some of the examples that we have. So for example, in our facility, we work a lot with groups that are working in phagocytosis or nanoparticle uptake or enzyme activity. So they do analysis of their samples at different time points. Like for example here, they put their cells and they will expose them to green nanoparticles during X amount of time. So you have here the median fluorescence intensity at 0 hours. So we start to plot time versus median fluorescence intensity to see how is the nanoparticle uptake of these cells. After 12 hours or 24 hours, you can start to monitorize how is this median fluorescence increasing. So that means that we have a clear nanoparticle uptake. This is another example. In this case, there are students that they were analyzing basically how is the cells affected by some particular drugs. The theory is for killing them. So in this case, there are students, and remember, students, they are able to kill everything. So basically we'll move from a very alive population to a dead population. And these are at different time points for the drug. As you can see, they are extremely effective. In this other case, it's just to show you an example at different time points of the kinetics for the cell cycle. So you have G0, G1 cells. Cells are in the double of DNA, but not uptake of the EDU. So EDU is coming only for the synthesis, so they will come around here. And finally, your G2M cells over here. So this is at 0 hours, 2 hours, 4 hours. So you can see how is the new population showing up here at the end of the synthesis down here. And again, it's really nice to monitorize what's going on onto your cells. What you have seen at the moment is basically me playing with PowerPoint and trying to put those slides one on top of the other. But there are some commercial softwares, like in this case Calusa, that basically is doing exactly the same thing in a more professional way. You put your samples that you have analyzed at different time points, and what you can do is put them all together and merge all this data as if we're just one file. So at the end, just merge them and you get one unique file. When you get this unique file, afterwards, what you can plot is time. Each one of these gaps here that you have in this time correspond to the separation between sample 1, sample 2, 3, 4. In this case, it's a kinetic study where you have your control at 1, 2, 4, 6, 10, 15, and 60 minutes. So in this case, we will keep red our control, and later on you will see the different populations moving as in blue. And you will see as time passes, you can start to see how these cells are reacting and which ones are physiological changes happening into this experiment. Then we have another assay here. This is quite simple. So in this case, we have a group that is working in fossils, and they want to make sure that they remove all the bacteria because they can degrade their samples. So they want to kill bacteria and make sure that they go at the lowest level. So basically, just run the sample at different time points, and you can see at the different time points we have a drop down on the number of events per microliter, which is quite simple. But in another cases, what we want to do is killing the cells and seeing that we are able to kill them. So again, here we just monitorize time, and you can see the drop down on our sample in just a few minutes. Let's go to another area. This one is called flow cytometry in situ. So that means that we have instruments, like in this case the Cytometry, and Cytometry is custom-made, kind of, but it's commercially available flow cytometer specialized in analysis of samples in the water. So you have kind of a normal flow cytometry for analysis of phytoplankton, and after the analysis, basically you just be able to take a picture of that particular event. So you can see at the same time the profile of the fluorescence, but also the image of each one of these events. So as you can see here, rather than just doing flow cytometry, we're doing imaging. So we're doing imaging flow cytometry, similar to what the image stream flow cyt are doing. So we can analyze all these parameters. And why they're interesting? Because we go from a taxonomic analysis of our sample population to something more taxonomical. Is that important? Yes. They are monitoring water, sea water or lake water, and this has biological outcome. The instrument is sit on the ship. In the ship, they can get the samples, move them into there, and that's fine. But it's better if we can get the samples a little bit under the water. So they put this instrument underwater, and they can analyze at different meters. So you can get it at 0, 24, 55, whatever meters you want to get the samples in, and analyze what's the flora and fauna on that deep. But even better, why have to be on the sea? If I can put the machine in a buoy, leave it there, and get the samples. Meanwhile, I'm in my office, taking my coffee, and analyzing the data whenever I want. So basically, they put this flow cytometer in the middle of the seawater, just put some solar panels to feed the batteries and everything, and then just satellite to send the information all around. What we will have now is all those samples that we are sending. We take all these images, but also we have the information about fluorescence, so we can get abundance, we can get the average cell size, we can get chlorophyll content, etc. And this will be versus time. we can make a further step and put this in correlation to things like the currents, the temperature in the water, the temperature side, the winds, etc. As they are doing, Gerard Gergerly is doing, basically just working on the dynamics of these seawater samples. So as you can see, we go for slow process, medium speed flow process, but we can also analyze processes that are really fast, that they happen really quickly into the cells. So when the people is working in this kind of process, mostly of them are working in confocal microscopy, taking images of their cells. So you can analyze the fluorescence intensity versus time at different time points. Now, the problem for microscopy is that you're working at 20, 30 cells, and statistically it's not quite representative. And that's why if you want to speed this up, you have automated microscopes, like the high content screening. In this case, you can put them in a box so they will be less scary, especially for your users. So basically, the machine is able to go to the same spot, take a picture, come back after a few minutes or after a few seconds and take more pictures, changing filters, etc., etc. So you can monitorize how is the movement of your samples. So you can do things like, in this case, is showing the kinetics that I showed before, but with the image right beside. So you can click on one particular cell and see which state and when they are changing. So it's not just the image. The importance of this is the amount of data that you can generate. And when you plot this information together, you can see in this case that there are some of the cells, like in this case, that they have a higher response to their stimulus. Meanwhile, in this other case, you have some other cells that they don't respond to this stimulus. They continue like if nothing has happened. Key question here is, you monitorize these kind of changes by flow cytometry. The answer is yes. I am not the first one. This is probably from the beginning of flow cytometry. One of the key studies was to measure the kinetics of cellular process. Obviously, there are some other people, like Elizabeth Simons, that they are the expertise into this area. So here is her showing up how to use the FAX 440, and she was analyzing samples around 4,000 cells per time point at 5 seconds intervals. And you can see here the data that she has got. As I said, I'm not the expert. We have Johnny Moore and Elizabeth Simons, that they are really the pioneers of the kinetics, and they know a lot, so it's fair to recognize what they have done for this field. So mostly of you, as I've said at the beginning, and mostly of us, we are working with pressurized systems. So you put the sample in, then the sample is pressurized, and we analyze the sample. So to do kinetics, what we have to follow up is mostly of the time with one method called stop flow method, where you get your sample in, you start to analyze a minimum baseline for X amount of time. Now you stop the sample acquisition, you download your sample, put your stimulus, and then put it back, get the same pressure than before, and continue with the analysis. All this requires some time, and this is time that we can lose some of the data, but at least the sample can be analyzed. This is the typical kinetic. This is shown in current protocols, and as you can see in this typical assay, what we don't know is how this population of cells is moving. Yes, this way, could be this way, or could be this other way. It's not just that the problem. In some software, you are quite limited in terms of the time that you have to show what is happening after 10 minutes. If you don't know, you cannot display that. The other option that you have is to stop your sample and acquire more sample afterwards. But again, you're losing sample. So in this case, it was a BD engineer, John Dunn, in 1991, was trying to make this a bit better for all of us. So he was modifying BD FAX Start Plus. With these modifications, you can see that he was able to do these kinetics without any gaps on them. Okay, taking this in consideration, another couple of BD engineers, they have developed what is called the time zero system. It's sold now by Cytec. So basically, it's an instrument that you can put in and move from kinetics like this ones with gaps to something like this without any gap, where you can see in a continuous what's going on after one particular stimuli. The limitation for this time zero system is just compatible with a few instruments that it will be not quite possible to get them now. So At this point, I want to say I have no commercial link and I have no economical benefit for what I will say about the Acuity. But I have been working quite a lot with Acuity and Ph.D. Since 2009. What I found is that they have an instrument that is quite interesting to do continuous measurement of flow cytometry, and this instrument is commercially available. And in 2010 we have published this paper describing how to do it, and this is what I will do now. What's the advantage of the Acuity system versus another flow cytometers that we have? It's basically the presence of the peristaltic pumps. The peristaltic pumps has the advantage versus the other ones that instead of the normal flow cytometry tubes that you have to make sure that it's sealing, and what we can use is appenders like this one. As you can see, what we can do now is we have a gap there where by using, in this case, we are using the chill loading tips, we can introduce any kind of stimuli that you want. We have plenty of space there, plenty of room to introduce whatever we are acquiring. So I will show you how we do it. So basically, we just get the sample is running at one particular time point, we just added our stimulus and we mix up. You will see that we are producing some air bubbles, which are great for mixing up and make sure that we have a very consistent and homogeneous staining. And as you can see, the SIP was always in constant contact with the sample, so we are continuously analyzing what's happening. So we move from this kind of analysis that we have done on the Beckman culture into this other analysis that we are doing now with the Acuity C6. So as you can see, we have no gaps on our analysis. Now, because it's a peristaltic pump, people have been asking for how much time we have to wait until we see when we add one particular stimulus and we see the analysis. So what we have done is basically added beads. The beads are something that is not physiological affected, so we just put the beads, and once we see it, we know how much time we have spent. So from the moment that we add them in, is here, and here is when we acquire them, is less than 3 seconds. So it's very fast, the analysis on this instrument. Before I continue, I will try to make some considerations, not just for kinetic assays on an Acuity C6, some considerations for any kind of kinetic assays. As I mentioned before, the first thing that you have to make sure when you're doing any kind of kinetics is to make sure that you have a very stable instrument. So you have to make sure that the instrument is clean, that you have stable flow, etc., etc. The other thing is that some of these experiments might be quite long, so you have to make sure that you are able to resuspend your cells, because if not, by the end of your analysis, you will end up in practically no cells. This is something that we have been able to sort out with this mixing system. Now, if you remember, we put some air bubbles. So how these air bubbles could affect our flow rate. So we did quick experiments. So we have our baseline. We added some extra cells here. So as you can see, there's no change. Later on, we added an empty pipette where we added some air and air bubbles. And as you can see, there are no change. And finally, what we did is basically add one stimulus to see that when we added the stimulus, something is happening. So we add, in this case, an ionophore, and you can see that something is happening to your cells. The other thing that we want to make sure is, as I said before, is that we have a continuous analysis. So you can see that we have kind of continuous, but it's not completely flat. We'll have some variation on the number of events per second. What we cannot see, or we shouldn't see, is this. It's so constant that it's telling you something about the instrument. And in this case, it's telling you that the C6 is saturated. So it's not able to resolve how many events per microliter you have properly. The other thing that you will see in this case is when we added the stimulus, and in this case you can see the gap here, an intentional gap here. You can see that after the addition of the stimuli, the number of cells that we have has dropped down. So if this drops down, that's normal, because sometimes you are killing the cells and they will start to blast and disappear from our analysis. Remember, if it's blasting, you will have some events. And this is what could happen. You have a higher number of events into your analysis because your cells are blasting. Now, is this affecting my sample? Yes, it will affect your sample, especially if you're working on the Acuity C6, because in the Acuity C6 we have a maximum number of events that we can accumulate in our well. It's 1 million events. So if you reach that number, just because, not of the cell, because of the number of particles or fragments that you get from your sample, it will automatically stop and you will not finish your experiment. So it's extremely important that you optimize your experiment, look at the number of events per second that you have, make sure that you have and you apply the right threshold based on the time of your experiment. What about the control on the additional time? Can you control that? And if you think it's necessary, you can control it. So what we will do is we will always have a baseline of 1 minute. We set up that limit to 1 minute. We can zoom in so we can be really detailed on the time of when we want to make this addition. So you can start to see the sample acquisition, the baseline. We are closer and now we have the pipette ready. It's already in the tube. We mix it up and we let the machine to work. So why is this important? It's important because now when you control the time, you can start to compare different cell lines or different stimuli and see which ones are reacting and how long they're taking them to react or when they're coming back to the baseline, if they came back to the baseline. So I will show you some applications of this real-time analysis. There are several ones. We will show some things about calcium, GFP, platelets, nanoparticle uptake, or staining optimization or apoptosis. Let's start with staining optimization. As you well know, time for the addition of one particular dye and optimize this addition is extremely important for experiments. For example, in this case, is a clear example of the site population, where the addition of the dye is extremely important. We have one group who was here working with us. They were analyzing the lipids into the algae just for the production of biofuels. So what the dye that they were using was BODyP. And when you look at the prospect, they are saying basically add it for a few seconds and then run it. How much is a few seconds? We want to make sure that they are on the plateau and that all the samples are analyzed exactly in the same way, that they are all exactly the same in the same way. So these are the kinetics of 4 different algae where at this particular time point, in this case we added BODyP and we wait to see when they arrive to the plateau of the staining. So you can see in some cases they go up and down, and then up again to the plateau. When we compare these 4 different cell lines, what you can easily see is that the plateau is reached at different time points. At least now we know when and how much time we have to wait until we analyze each one of our samples. We have been working as well with platelets. We analyze time in this case versus forward scatter, side scatter, annexin 5, C41A, and we can analyze and compare each one of them. In this case we have 2 stimuli. One of them is clearly shown by a gap. The other one you cannot see, but we know exactly where we have the stimuli. You can see how it's bending down over here. You can see that it's going down in annexin 5, but at the same time 41A is going up. So there are several things happening. If you look after this gap, you can see a drop down on this point. If we don't have an instrument like the Acuity and we spend slightly more time, you will not see that kind of drop down on the data. So rather than this, it's better to see this drop down in the full. We have done experiments about erythrocytes and how we lyse them, and you can see here our erythrocytes in forward and side scatter, and you can see that by the addition of distilled water we started to drop down in both parameters. Now, is this a real thing, or is because we added distilled water that is affecting just basically our instrument or the measurement of our instrument? So what we added some internal controls. In this case we added some beads, and we did the same experiment adding distilled water at different time points, and you can see all the time that the changes on the forward and side scatter on our erythrocytes is not affecting the forward and side scatter for our beads. So what we have is a real change. We have nanoparticle uptakes. In this, as I mentioned before, we have groups interested on the nanoparticles and the effect of these nanoparticles into the cells. So they put the sample in, these are the cells, and now we put the nanoparticles in. Suddenly what we saw in a few seconds is that they have a huge amount of nanoparticle uptake. Is this a real one or is it a fake one? So one of the things that we will try is to control the temperature, because we can do at 4 degrees, so it shouldn't get enough nanoparticle uptake, or we can increase the temperature. If we increase the temperature, we should get from 24 room temperature to 37, we should get a higher phagocytosis or nanoparticle uptake. So what we did was put our sample just basically in a water bath, controlling the temperature and doing that experiment. If we want to cool them down, that's quite simple. Basically what we put is our sample in the middle of ice. Okay, this is quite rudimentary. We could do it a bit better. So in this case, basically what we have done is remove the tube holder. We have, in this case, a thermoshaker. We put our sample in, and we can still move the sample and stimulus inside the sample. As I said, it's a thermoshaker, so we can control the temperature, but also we can keep the samples shaking. So that means that we will have all our cells constantly in suspension. So when we compare this analysis with the nanoparticles, you can see the baseline and how this increased at 2 different time points where we have 2 different additions of our nanoparticles. With 37 degrees, as I explained before, we expected a higher increase on the nanoparticle uptake, and this is not happening. When we put them on ice, we expected to have no nanoparticle uptake, and again, this was not happening. So basically what happened is that rather than nanoparticle uptake, what we have is the nanoparticles attached to the surface of the cells, and this is what we were measuring. If we are talking about calcium, that's something we've mentioned before. We are talking about fragmented cells, so that's a way of removing these fragmented cells from our analysis. The other thing is that we have to verify everything. So are you sure that these low fluorescent events are fragments, or they are not cells? So we add other dyes, like for example DRAG5, so nucleated cells, so you can see forward and side scatter, and on the other side we have the fragments without any DNA dye. What you can see is that the ones are with the DRAG5 here, no DRAG5 there, and they correspond to the top fluorescence or the lower fluorescence intensity. In most of the cases, we always recommend users to remove the dead cells from the analysis because they will highly affect the final data and your results. So in this case, I will show you this gating strategy from a group. They were asking me how to do the analysis of these kinetics from University of Cape Town. So you can see here fluorescence versus propionio line. So we will exclude the dead cells from the analysis. We'll work on the singlets only, and you can see if we don't include these 2 populations, if we don't exclude the dead cells or if we don't exclude the aggregates, you can easily see 1, 2, and 3 different subpopulations in forward and side scatter. If you do this gating strategy, it's a big difference of what we will analyze, and this will reflect clearly on how your calcium is analyzed. Okay, this is before, this is after this gating. So remove your dead cells from your analysis. The other thing is how many profiles you can see. You have different profiles, so in this case we have samples that they're not affected by any calcium or any stimuli. You can see some others that they go up and they continue up, and other ones that they will go up in the calcium and then they will come back. You will see samples that when you add them to a stimulus, half of them are going up and another portion of them they will have no reaction, as we saw as well previously in the high content screening. We have a mixed population of cells that you add one stimuli and, oh, surprise, they come back to the same baseline. We have 2 that they come back to the same baseline, but it's between the first and the second one. So it's quite interesting area. This is another thing. We can do calcium flux and transfected cell lines. So you have here a wild type, and you can see the profile in calcium, and here you have transfected cell lines. Transfected cell line that you can see here, this is your non-transfected. Here is non-transfected and transfected cell line. So if we gate on the non-transfected, you can see that their profile compared to the wild type is quite similar. But when we look at the profile of the transfected M-cherry samples, you can easily see that this is completely different to the wild type or non-transfected ones. As I said, it's not just fluorescence what we can measure. We can measure as well changes, as you can see here, changes on the forward scatter, how my forward scatter is changing, or the presence of these fragments on this side here. These are several parameters that we can analyze at the same time. So in this case, we have our sample, and you can see that there is a slight fluctuation here. The same here, this is slightly fluctuation. Is it a real fluctuation due to my experiment, or is it a real fluctuation based on the instability of my instrument? We have been sure that the instrument was stable before we started. How we can make sure that it's stable? Well, we added some beads to monitorize how stable is our analysis all the time. So this is an internal control that we have in our analysis. You can also exclude them because we don't need them, and just basically gate out all these beads afterwards. But this is a nice control. So as I said, in this case we are working with mitosocs. We have drag 7. In this case we have forward scatter and side scatter. You can see here that we have several actions after one stimuli. If we want to compare them, which one is coming first, which one is coming afterwards, what we will try is to do an overlay between all of them. But how we can overlay this population here, there is clearly a mix. When you try to do the overlay with the median fluorescence intensity, you can clearly see that the median fluorescence intensity in this population is not clearly showing what is happening. So we'll discard this one and we'll keep this one as our baseline. And then we'll start to overlap with forward scatter. We can start to look at which one is happening first or afterwards. Let's go for the last part of the talk. I will be super quick now. So this is an actual project that we have with a company called Babrima, that they have this instrument called mitoplicator, that they're using an antenna to generate nanosecond pulse of electrical fields. And the idea for them was trying to kill bacteria, trying to kill cells. But what they found out is the opposite effect. Rather than decreasing the number of cells, with the treatment, they found out is that they have a higher number of cells after a few days. The cells were proliferating much faster. So they thought, okay, that's ridiculous, that's not good, because we want to make money. But suddenly they understood that there's something completely different. We can make money if we sell this to the pharma companies, because then they can have more cells in less time. But they don't know what is happening. So this is basically what they told me about their instrument. So we have an antenna, we put our cells with this electrical field, and what we are producing is these pores on the membrane. And what we will see is changes on the calcium. And that's why they were in contact with me. They wanted to measure how is the calcium affected by this antenna or this instrument. So the initial idea was to put the acuity on top of the mitoplicator and get the samples directly measured from the mitoplicator. But we face a couple of problems here. The first one is that this is the area that we have to put our sample in, and this will produce a big electrical field. And that's why we have this plastic here, because it has to be on top of this one to be able to avoid any electrical charge. And I don't want to burn my flow cytometer, if not I would be in trouble. So how we ended up doing this? So we put a couple of tubes, silicone tubes similar to the ones that we use in the science or the hypersite, and one of them will be to collect the sample and move them directly into the acuity. The other one that we have here is to introduce our stimuli or just to keep the cells in suspension. First thing that we have done here is measure the stability of the instrument. You can see this is the stability on the normal holder, and this is in the instrument. As you can see, it was not quite stable at the beginning, but after a while it becomes really stable. So we try to improve the system, and as you can see here on this picture, we have a peristaltic pump between the instrument, the mitoplicator, and the acuity. We have improved the time by shortening down these ones, and now we have a delay of 15 seconds, and I'm sure we can do it much better. What we have measured things like CFSE to see the proliferation of our cells, flow 4 to measure the calcium, drag 7 to look at the viability, mitosocs to measure oxidative stress, body P for the production of lipids, and GFP as a measurement to make sure how is the protein content of the sample stable and still inside the sample. We have been working with several cell lines, the C6, the HEX, CTO, URCATs, or even platelets. If you remember, we have done some experiments before with M-Cherry, non-sorted, and the other ones sorted. And you can see in this case they are quite similar, but quite similar is not the same than the same. You can see that in this case the control has slightly more time to come back to the baseline than the transfected. And again, it's slightly more, but we want to measure that difference. And there's no real parameters to measure that difference. And this is where I have to thank a lot the Isaac Scholar program to put me in contact and allow more collaborations between Isaac Scholars. And in this case is with one of the Isaac Scholars, Kerry Toldi. He's based in Budapest. He has developed software called FACSkin, which is basically used for the kinetics analysis of calcium. It's a free software, free to use, and I'm sure he will be delighted to help you with everything about how to use their software. So what you can get from this software is basically the beginning. You have the starting value, the slope at the first 50%, which one is the maximum value, how much time or which one is the slope at the 50%, how much time you will spend to arrive to this value, and which one is the final fluorescence intensity that you get. So as you can see, we can translate our more or less to real data. The software is quite simple to use. You have forward scatter, side scatter. You can get into your samples, look at things like, for example, in this case, the drag7. How is the drag7 affected? Because it was one of the parameters. Or you can basically gate out the dead cells, looking only on your alive cells, and do the kinetics of calcium only on your alive cells. And you can do it in fluorophore, or you can do it as well on forward or side scatter. And you can do multiple gates to look at the reaction of your cells before the stimuli, after the second stimuli, etc., etc. So all of these can be analyzed, and all of these will be transformed into graphs like this one. You see here, mitoplicator off, mitoplicator on, mitoplicator on and off for the drag7. And all of these, as I said, can be transferred into data that we can easily put into graphs that everybody can understand. So in this case, mitoplicator blue positive, if negative is the orange one. So you can see that there's a real difference in this case for the C6 glial cells. Not all the cells will react in the same way. As you can see, when we try the same experiment with the HEC, we find out that this relationship is slightly opposite. So we need to keep going with this project. We want to keep going and understanding what this instrument is doing. And I'm sure the company is also interested in keep going with this collaboration. So this is an ongoing collaboration that we have between Prima, several groups in the university, Fluxion, BD Acri, and FACSkin. I think you have understand a little bit more why it's important to use time, why it's important to do this analysis. To be honest, I think time is a free parameter that nobody is using, or practically nobody is using, and it's worth it to consider for the future. I just want to end up thanking, acknowledging everybody that has been working with me and make this tutorial possible, starting with people like Gethin Bing and Alice Vines, helping at the beginning with the development of this application that we have done with the Acri. We have Chava, Anna, Esther, Gary Litoldi from the group in Budapest. We have the group here. It's Pabrima. We have Esther over here. She has been a key piece on this final project. And I want to thank, obviously, PD, Acri. They have been extremely supportive to this project. Fluxion, the same. And finally, all the Isaac Scholar program. And finally, I'd like to ask everybody to participate into the Cyto 2015 that will happen in Glasgow in June 2015.
Great, thanks very much, Alfonso. That's an excellent presentation. So we have a few questions. If anyone else has a question, please feel free to post it in the questions box that appear on the right of your screen, and I'll be happy to post them to Alfonso. So Alfonso, are there instruments other than the Accuracy 6 which allow this type of continuous flow cytometry?
Yes, as far as I know, the new, don't know exactly the name now, for the cytogene that has been developed by the Chinese company that has been recently bought by Beckman Coulter. Basically, it's an Acri, it's a modified Acri, what they have done. The Jack Ball and his group has developed another Acri, a different version of the Acri. Cannot be sold as Acri, but it's quite similar, I think. And I haven't tried yet the ASIA, the Novocytes from ASIA. I think these are the ones that you can do this kind of assay.
So there may be others. Good.
Yeah.
On the C6, what's the maximum measurable time that's possible?
That's a good one because you can analyze 19 days, 26 hours, 24 hours, and I go. So it's great for having big students that they don't care to be in the lab for many hours and days.
They don't need to sleep.
They don't need to be there. Now, having said that, it's quite interesting because the maximum number of events that you can acquire is 1 million events per well. And this is a big limitation on the instrument. I asked several times BD to take that out and leave the user to decide between having 1 million events or having 10 million events per well. Because in this kinetic assays, it would be fantastic to have this option of opening it to the number of events that you want.
That would be a nice extra feature, really, wouldn't it?
Yeah, definitely.
Where you're performing these kinetic studies where you're controlling the temperature, I know some of these instruments have like 96 well plate loaders and that. Is it possible to control the temperature in this situation?
Yeah, this one person is called Chris Jones. He's based in the University of Reading in UK. He has presented together with me in a BD webinar a couple of weeks ago how to do it. Basically what he's using is using the heating plate that you use for keeping the animals warm during the surgery, like mice or rats. So it's really flat, has exactly the same size that you can put underneath any 96 well plate, so you can keep your sample warm all the time, warm or at the temperature that you want. So it's really nice, and I'm sure he can give you more details. It's Christopher Jones from University of Reading in UK. He has done it for platelets. He's working mainly in platelets, and you can see his data. It's amazing.
We'll have to look that up. Thank you. And just quick last question. Are you aware of any other applications or groups that are actually using this type of methodology?
As I said, this is open for the imagination. We have recently published with Frederick Hams in Switzerland paper. It's in +1, and they are working with water to measure the quality of the water, things like the water in the bottles, and they're working a lot in bacteria, so they have several applications showing how to kill bacteria or how to monitorize bacteria, how the bacteria is affected by things like temperature or light and things like that. So they have shown quite a few examples of bacteria analysis using this kind of approach.
Someone has suggested that the attune.
Well, the attune,
maybe also.
the attune is not the right one. I was fascinated with the attune. I love the attune, but I'm afraid it's not possible to do it with the attune, because the attune, although you can put an Eppendorf, the problem for the attune is an injection. So you will get the sample into one syringe, and once it's in the syringe, they will start to pull the sample in.
Ah, yes.
If you want to do the stimuli, what you will do is take out the sample from the syringe, put it back to the Eppendorf. Now you put your stimuli in, and then you suck it up again, put it back into the syringe and push it. So it will be again a stop flow method. But I was thinking to do it as well with the attune, but then I found out that cannot be possible.
Okay, just one of those limitations. And then lastly, we just have a comment here. The very interesting dynamics of calcium flux suggests that gene activation and the impact of a drug or nutrient could be used to measure the time and amount of cellular synthesis of a target cell culture in the multiple well-heated plate. So really it's just a question on that maybe it might be a good system having it on a well plate, I guess, like a high content screening system where we can track cellular synthesis over time.
It's what I'm missing, because it would be great to do similar things with imaging cytometry. As far as I know, the AMNIS is again another instrument that could be great for doing this kind of analysis, but the way the sample is delivered is limited. I was talking with David Basili about that, but at the moment they have technical limitations to do it that way.
So stay tuned.
Yeah.
A last question here, what are stains that are used to assess viability? And I just really direct that question just to look up the webinar from yesterday, which was relating to tracking cell cycle analysis by flow, and really Derek Davies' talk there covered a lot of the dyes that are important for that. So I think it's really beyond the scope for today. So that just brings us to the end of the seminar. So I'd like to thank you again, Alfonso, for a very illuminating presentation and a great discussion.
Thank you, Murray.
Finally, thanks to you, the audience, for taking the time to attend and to listen in. If you've enjoyed this seminar and would like to view the video recording of this session, please visit the seminars page on bitesizebio.com. It should be available within the next 24 hours. There you can also see the other webinars we've lined up for you in Bite Size Bio's Webinar Festival. So until next time, good luck in your research and goodbye from all of us at Bite Size Bio.